I'm going to focus on the challenges for ethics and for public policy posed by synthetic biology. And I'm going to do that by the device of proposing a couple of conceptual frameworks for thinking about synthetic biology, ethics, and public policy. And the frameworks, at least as I'm thinking of them now, are not mutually exclusive. They might in fact be complementary. You might even convince me that they're duplicative, but we'll find out. In the spirit of the Hastings Center's work, we have three mantras, only one of which I'll mention in a moment, and that is the one that says good ethics begins with good facts. So it's important to know what the phenomenon is about which you're presuming to opine. And so I will begin the formal talk with a little discussion of what synthetic biology is, at least as we've come to understand it. Now I have three goals for my remarks tonight. One is to provide a brief description of synthetic biology's main streams of work, because one thing that immediately becomes apparent is synthetic biology is not one thing. There's several different kinds of work that may or may not converge. Secondly, I will employ mostly familiar distinctions that are meant to help sort through the benefits, risks, and policy options. Most of this will sound quite familiar to you. And thirdly, I'll describe these conceptual frameworks and begin the process of identifying the challenges that synthetic biology confronts and the central questions that will have to be addressed for us to really have a more satisfying understanding. I should note that the Hastings Center is a rather small and desperately underfunded research institute. We were founded on a shoestring 40 years ago. Today, we are running on a shoestring. It is the longest shoestring that I'm aware of in these U.S. research institutes. But we do work as teams, and so I have to want to express my gratitude both to my colleagues who are also trying to clarify our thinking on synthetic biology, and I will credit them when I'm using their ideas. But I particularly want to thank my research assistants. We have two young research assistants without whom this PowerPoint would more resemble a kindergartner's blotted copybook than whatever polish you may see in it. So we began with trying to get some definitions of synthetic biology, broadly understood as the deliberate design of novel biological systems and organisms. It draws on principles elucidated by biologists, chemists, physicists, and engineers. And then comes the kicker. In essence, it is about redesigning life. And since we're convened at the Royal Society, I thought it appropriate to quote that. This group, known more conventionally as the ETC group in Canada, has been a critic of genetic technology generally, but they offer a fairly neutral and I think reasonably accurate definition: design and construction of new biological parts, devices, and systems that do not exist in the natural world, and the redesign of existing biological systems to perform specific tasks. And the European Commission definition, I won't bore you by reading it also, but it's reasonably consistent with the ones you've just read. Now, there are some examples of what are claimed to be goals of synthetic biology, and it's important to bear in mind that most of what we talk about is aspirational in terms of the science and technology, not so much genuine accomplishment. There are some, but a lot of it is aspirational. So, industrial scale chemical production, using re-engineered biological cells to produce biofuel for engines, for example. Creating new strains of Pseudomonas to degrade pesticides to use for environmental cleanup. Ways of this one is not quite so hypothetical. This is being done, but not at commercial scale. Redesigning a bacterium to produce artemisinin, which is a critical antimalarial drug from simple sugars. This bug has been designed. Now, I want to thank Professor O'Malley for co-authors for a terrific paper that helps sort out the different streams of synthetic biology. Her work identifies three major streams. What I'm going to describe to you is our adaptation of that line of work. So one is minimal cell creation. That is, making the smallest possible cell. Some investigators, for example, Craig Bender, talks about designing a chassis, like the analogy to an automotive, to the car. So you create a chassis, then you build a car upon that chassis. So the idea would be to create some minimally functional cell into which you could put the components you wanted to accomplish whatever tasks you wanted. So you could build it into a delivery van, into a sports car, whatever it was that you were attempting to design. That's one stream. The second stream would be DNA-based device construction. This draws very heavily on the engineering mindset that has pervaded synthetic biology. And the third would be a kind of advanced, ultra-sophisticated genetic engineering, doing what genetic engineering has been doing for decades, but in a more decisive and successful and targeted way. So the first, the minimal cell, again, that little beautiful little bit of animation there, totally Polo's work. You can do it either top-down or bottom-up. So in the top-down version, you take a very small cell, and that's a picture of the carcinella genome. It's one of the smallest known genomes of any living organism. And it's an endosymbiont, that is, it's not free-living. It relies on its environment, the insect environment, to survive. What you do is you take a genome like that and you knock out bits of it. And you keep knocking out bits of it to see how far you could go and still have a functional cell. And this has been tried a number of ways. One of the ironies here is it turns out that you can go different pathways and you might still end up with a viable cell. So for some have used some scientists have proposed the analogy of the four-legged stool. Well, you can knock out any one of the four legs and still have, the three remaining, still have something that's usable as a stool. And in a way, it doesn't matter which of the four legs you knock out. So it turns out you knock out some genes and you might still get a functional cell, but you leave those in and knock out some other ones, and you might again still get a functional cell that will just function differently. So biological systems are complex in ways that stools are not. The bottom-up version of this strategy would be literally to take and to string together bits of DNA to create synthetically an artificial minimal cell, component by component. And different approaches are taken by different investigators, depending on whether you regard him as famous and notorious, Craig Venter, George Church, and others. Major stream number 2, DNA-based device construction, really is an engineering mentality applied to biology. Here you have the steps of standardization where you're trying to define particular parts that are known colloquially as biobricks. So you take it, you might be bits of DNA or RNA, bits of protein, putting them together, and you have a construct that accomplishes a particular task. It might, for example, sense a certain level of glucose in a fluid solution, and then another biobrick would then be attached to that one, and that one might send a signal to turn on a process. A third biobrick conceivably could then produce insulin. That's the concept, right? So you'd actually have these definable components, these biobricks, discrete parts, in the same way that people who understand electronics have transistors and other kinds of circuits and switches and things, that they put them together in certain ways to accomplish particular tasks. That's the idea behind